we're going to continue talking about protein folding and how proteins fold in 3 dimensions. We're not going to spend a lot of time on this section, but we will spend a little bit. So what I want to emphasize here is that there are many different ways of describing a protein molecule to other people. And traditionally, there are 4 main ways of describing a protein molecule. So imagine that I had a protein molecule that I was interested in and I wanted to describe it to you. There are 4 main ways that chemists and biologists usually talk about. They are called primary, secondary, tertiary, and quaternary structure. And they are abbreviated, 1 with a little degree symbol, that means primary structure, 2 with a degree symbol, 3 with a degree, or 4 with a degree. So I want you to sort of become accustomed to sort of the abbreviation. Now, I haven't explained what these different ways of describing a protein are. I'm going to spend most of my time talking about primary, not very much time talking about secondary, a little bit of time on tertiary, and not much time on quaternary. So we're going to focus mostly on primary and tertiary. So, different ways of describing a protein molecule. The first is primary structure. Another way of thinking about what primary structure is just listing the order of the amino acids in your protein molecule and how they are attached to each other. And that's all you're talking about when you're talking about primary structure of a protein. So, as an example, suppose I had a protein molecule and I didn't want to tell you anything about how it actually folded up in 3 dimensions. All I wanted to tell you is which amino acids were connected in which order. And so I would come up to you and say, look, I'm working on a certain protein and the order of the amino acids is alanine, alanine, leucine, isoleucine, proline, methionine, etc., etc. If I did that and I didn't describe anything about the 3-dimensional shape of this protein molecule, you would say that I am talking about the primary structure of the protein. Another way of saying it is I'm talking about the amino acid sequence of my protein molecule. And that's pretty much it. The times when you would want to talk about only the primary structure are times when you don't really care about the 3-dimensional shape, or describing the 3-dimensional shape is just going to confuse things for people. An example of that you've already seen. So when you're talking about primary structure, it doesn't tell you anything about the 3-dimensional shape of the protein. But you have seen an example of that, and that's shown here. This entire block here is the primary structure of hemoglobin beta. Hemoglobin beta is part of the hemoglobin molecule. And so this is the order of the amino acids in a protein molecule called hemoglobin beta. It starts with methionine, then valine, then histidine, and on and on and on, and then you go to line number 2, and basically all you are seeing is the order of the amino acids and how they're attached. The amino terminus is here, the carboxy terminus is here. I'm not telling you a single thing about the 3-dimensional shape of this, but I am telling you the order of the amino acids. Why do people bother to do this? Well, they do it because it's really easy to point out where changes take place in the chain. You can see here highlighted in red, this is the glutamic acid amino acid that changes when people have sickle cell disease. So a lot of times, if I showed you the 3-dimensional shape of this particular protein molecule, it might just be confusing when all I really want to do is point out to you that this is the amino acid that changes in people who have sickle cell disease, and this amino acid shows up 1, 2, 3, 4, 5, 6, 7 amino acids from the amino terminus of hemoglobin beta. And so it's an easy way of pointing out to people where certain changes take place, where certain mutations take place that can cause certain diseases. So that's primary structure of a protein molecule, when you just list the order of the amino acids in your protein molecule. There's something called secondary structure. We're not going to spend a lot of time on this. This is basically the 3-dimensional shape of short pieces of the protein molecule. So you could see here there was a little spiral. If you were talking about only this little spiral, you would be talking about part of the secondary structure of this entire protein molecule, or this spiral over here if you were talking about that spiral, which is formally known as an alpha helix, but you don't need to know that. If you were talking about that helix, you would be talking about the secondary structure, a little piece of secondary structure of your protein. And these little twists are called turns, and there are many different kinds of turns. So anytime you're talking about a little piece of the three-dimensional shape of a protein molecule, but not the whole protein molecule, then you're talking about secondary structure. And there are many different names, but you don't need to know them. There's alpha helix, beta sheet. There are many different types of turns. Don't worry about that. Tertiary structure, I do want you to know what it means. That's the sort of overall three-dimensional shape of one chain of amino acids, of one protein molecule. So if I was interested in sort of describing the entire three-dimensional shape of this particular protein, then I would be talking about its tertiary structure. So again, primary structure, you don't care about the three-dimensional shape, you only care about the order of the amino acids. Tertiary structure, you're talking about the overall three-dimensional shape of one particular kind of protein. Quaternary structure is sort of a special instance. Don't worry about anything here. All you really need to know is that sometimes chains of protein molecules will attach to each other to make even larger molecules. And when multiple protein chains of amino acids connect to each other, then you say that the larger molecule has quaternary structure. But again, I only care that you know what primary and tertiary structure are. Protein unfolding. So we've said that proteins need to fold in order to do their jobs properly, but proteins can unfold under certain environmental conditions. I want you to sort of think of proteins as being like prima donnas. They're like the hard-to-deal-with actor on the Hollywood set who won't come out of their trailer unless they're given, Evian sparkling water and only green-colored M&Ms, and they're constantly complaining about how they can't work under the conditions that they're being subjected to. Proteins are a little bit like that. They need the right temperature, they need the right pH, they need the right salt concentrations in order to do their jobs. If you deviate too much from the temperature that they expect to work under, or the pH that they expect to work under, if you deviate too much, the proteins unfold in 3 dimensions and they don't do their jobs. They basically fall apart if you deviate too much from the environmental conditions that they're accustomed to. This unfolding of proteins in 3 dimensions has a formal name. It's called denaturation. So denaturation basically means when a protein unfolds from its normal functional 3-dimensional shape. And denaturation comes from the fact that de kind of means away from, and denaturation means moving the protein away from its natural state, moving it into a state where it's not folded up properly. And this almost always happens when you put the protein into an environmental condition that it's not accustomed to. You put the protein at a temperature that's either too hot or too cold, or put it at a pH that's too acidic or too basic. So that's protein unfolding in a nutshell. I want you to know what denaturation means. This is a real-life example of protein folding or protein denaturation. If you have ever made sunny-side-up eggs, when you crack the egg and put it in your frying pan, the liquid that's surrounding the yolk is clear. And eventually you heat it up and the liquid sort of turns white like this. What's really happening in that case is when the liquid is clear, there are a bunch of proteins dissolved in that liquid. They're folded up nicely in their 3-dimensional shape. However, what you do, and once they're folded up, they're nicely dissolved in the liquid. What you do, however, is you heat up the pan. And you heat up the pan to the point where those proteins are no longer accustomed to being folded and working properly at the very high temperature of the pan. They're proteins from a chicken, and the chicken lives at pretty much the temperature that we live at. Its body temperature is pretty similar to ours, and so its proteins are accustomed to folding up properly and working properly at the temperatures at our body temperature, basically. And what you do, though, is you heat up those protein molecules to a much higher temperature. Those proteins aren't accustomed to folding up properly in the pan at that very high temperature, and they all pretty much unfold. When they unfold, they expose their hydrophobic amino acids to each other. The hydrophobic amino acids don't really want to be around the water that is in this liquid, and so they end up slamming into each other in an attempt to bury their hydrophobic amino acids again. If enough of them basically connect to each other, if enough protein molecules connect to each other, they don't stay dissolved in the liquid, and they essentially undissolve from the liquid, and they come out of the liquid, and they cause the liquid to turn white. Because they're not folding properly, they're sort of slamming into each other, and they precipitate out of the liquid. And so that's essentially what's happening when your clear liquid in the sunny side up egg turns white. You're unfolding the proteins, forcing them out of the liquid and forcing them to turn this liquid into a white color. So you are denaturing proteins when you heat up the eggs in this way.